Now students, in this lecture we are going to have discussion on cell wall of gram-negative bacteria. The cell wall of gram-negative bacteria is more complex as compared to that of gram-positive bacteria. There is only 10% of peptidoglycan that is present in the cell wall of gram-negative bacteria as represented in the figure. This peptidoglycan content is less as compared to that of this gram-positive bacteria. So the bacteria is composed of outer membrane which lies outside this thin peptidoglycan layer. So it contains this outer membrane, this upper blue color that is representing the outer membrane which is outside this thin peptidoglycan layer. This bacteria also contains the protein which is known as Gros protein. Gros protein is basically an anchor protein that anchors the outer membrane. This is the outer membrane. This anchors this outer membrane of gram-negative bacteria to peptidoglycan layer. So, what is periplasm which is present in bacteria? This periplasm is actually the region between the cytoplasmic membrane and the outer membrane. This outer membrane. So, this is the region between this outer membrane and the cytoplasmic membrane, and this is filled with a gel-like fluid. So, this is called the periplasm. So, in gram-negative bacteria, there are proteins which are present in the periplasm, and these proteins have ability to be translocated, to be transferred across the outer membrane. Now, when these proteins are in the outer membrane here, then we will call them as surface proteins. So periplasm is actually the fluid that is, it is the space which is filled with the proteins that is involved in various cellular activities, including the nutrient degradation and transport. So until now we have discussed about the periplasm, that this is the region between the cytoplasmic membrane and the outer membrane is filled with a gel-like fluid, and this fluid is actually the periplasm. And this is very important. It contains the proteins, and these proteins have ability to be translocated to the outer membrane, and at the outer membrane we call these proteins as the surface proteins. Now, this periplasm is filled with the proteins that are involved in cellular activities including nutrient degradation and transport. Then we have outer membrane in gram-negative bacteria. The peptidoglycan layer is actually, this peptidoglycan layer is surrounded by the membrane in the gram-negative bacteria, which is the outer membrane. Now, its outside is actually composed of the LPS, that is lipopolysaccharide rather than lipophospholipase. So, on the outer side we have LPS, we have lipopolysaccharides. So, for this reason, the outer membrane is also known as lipopolysaccharide layer, that is LPS layer. This figure is representing, this region is representing that we have LPS in the bacteria, we have LPS in the gram-negative bacteria. So the outer membrane functions as protective barrier and it excludes many toxic compounds. So this lipopolysaccharide is very important medically. It is composed of 3 parts, while among them the 2 parts are very important. These 3 parts are lipid A, we have core, we have O antigen. This lipid A is embedded in membrane, while the core, which is a polysaccharide on the surface of the membrane, the O antigen, this O antigen, which are the short polysaccharides, are extended out from the core. So this here in this figure, this region is representing the lipopolysaccharides. Here we have lipid, then lipid A, then we have core, then we have O antigen, that is also known as O polysaccharide. Now, what is lipid A? Lipid A plays a significant role in our body. It plays a significant role by which our body can recognize that I have any external agent which is present in me. It plays a significant role in our body's ability to recognize the presence of invading bacteria. This is toxic. This is toxic in nature. As a result, they act as endotoxins. These causes symptoms like fever, diarrhea, and shock. So then we have O antigen. It is composed of carbohydrates, including glucose, lactose, mannose, and this O antigen can react with other specific antibodies. So students, this was all about the cell wall of gram-negative bacteria.